Have you ever looked at a chromosome and wondered how scientists actually create a map for it? I mean, not just a blurry picture, but a really precise chart that shows exactly where every single gene lives? How on earth do they measure those tiny, invisible distances? Well, the answer is surprisingly cool, and today we're going to walk through the whole process step by step and build a real genetic map from some raw lab data.
So here's our game plan. First, we'll set up our mapping quest. Then we'll learn how to read the genetic clues, the different data types we'll be working with. After that, we'll reveal the formula that turns these clues into actual distances. We'll use it to find two different genes, and then for the grand finale, we'll put it all together to build our very own chromosome map. Let's get started.
All right, welcome to our gene mapping quest. The challenge sounds pretty straightforward. Take a list of numbers from a genetic experiment and turn it into a visual map of a chromosome. Think of us as genetic detectives, piecing together clues to uncover a hidden biological blueprint. So, how do we actually pull this off? How do we find a gene-specific address on a chromosome? Well, the secret lies in something called genetic recombination. You know, that shuffling of genes that happens during meiosis. It turns out, by counting how often this shuffling happens between genes, we can actually calculate the distance between them.
Okay, before we can crunch any numbers, we first have to understand our clues. In this kind of genetic cross, our results come in these neat little packages of 4 reproductive cells, or spores, and these packages are called tetrads. They can be sorted into 3 distinct types, and each type tells us a different story about how the genes got shuffled around.
First up is the parental ditatype, or PD for short. This one's the simplest outcome. It basically means no shuffling, no recombination happened between the genes we're looking at. All the spores end up looking exactly like the original parents. This is our baseline, our starting point.
Next, we have the non-parental distype, or NPD. And this is pretty much the exact opposite of the PD. This outcome only happens when a very specific kind of gene shuffling goes on, and it results in spores that are all different from the original parents. So when we see an NPD, it's a huge red flag that recombination definitely happened.
And finally, there's the tetratype, or T. This one's a mix. You get 2 spores that look like the parents, and 2 that are recombinant, or shuffled. This result also tells us that recombination happened. So keep this in mind. Both NPD and T tetrads are key indicators of genetic shuffling. All right, here's our raw data from the experiment. As you can see, we found 6 different classes of tetrads. The table shows us how many we counted for each class and what type it is. So for example, class 1 is a parental ditype, and we found 15 of them. Class 2 is a non-parental ditype, and look, we only found 1. Then classes 3 through 6 are all different kinds of tetratypes. If you add up all those counts, we get a total of 49 tetrads. This table has every clue we need to solve our puzzle.
Now that we understand our clues, we need the right tool to make sense of them. And luckily, there's a single formula that lets us convert all these raw counts into a precise genetic distance. Let's check it out.
And here it is. I know, it might look a little intense, but it's actually just a simple ratio. On top, in the numerator, we're basically counting all the recombinant events. We take all of our NPDs, and then we add half of our tetratypes. Why half? Well, because in a tetratype, exactly half of the spores are recombinant. Then, we just divide that by the total number of tetrads we counted. That gives us the frequency of recombination, and that percentage is the key to our entire map.
Okay, time to put this formula to work. First up, we're going to calculate the distance between a gene called PAN and a fixed point on the chromosome known as the centromere.
The process here is really straightforward. We'll just grab the right values from our data table for the PAN gene, add up the tetratypes that matter, plug everything into our formula, and boom, we'll calculate the final result. Let's do it. Okay, let's pull the numbers for the PAN gene. Looking at our data, for the PAN gene's distance to the centromere, the parental ditatype count is 15. Now this is important. None of the tetrads count as an NPD for this specific calculation, so that value is 0. The tetratypes that are relevant to PAN come from classes 4, 5, and 6, and if you add those up, you get 32, and our total number of tetrads is still 49.
So now we just plug those numbers right into our formula. We've got 0 for NPD, plus 1/2 of our 32 tetratypes, all divided by the grand total of 49. Then we multiply by 100 to get our percentage. And the answer is 32.7%. So what does that number actually mean? Well, here's the cool part. That percentage directly translates into map distance.
We can now say, with confidence, that the PAN gene is located 32.7 map units away from the centromere. We've just placed our first pin on the map.
Awesome. See? One gene down, one to go. Now we're just going to repeat the exact same process for our second gene, which is called PDX. The method is identical, but as you'll see, the numbers from our data are going to be a little different this time. Right, let's pull the numbers for the PDX gene's distance to the centromere. The parental distype count is still 15, but look, this time we do have an NPD. It's that single one from class 2, and the tetratype classes that matter for PDX add up to 16. The total, of course, is still 49. These new values are going to give us a totally different result.
Plugging these new values in, we get 1 for NPD plus half of our 16 tetratypes, all divided by 49. So let's run that calculation. And that calculation gives us 16.4%. Just like before, this means the PDX gene is 16.4 map units away from the centromere. We've just found our second location. Okay, we've done all the heavy lifting. We've calculated 2 super important distances. Now, for the really satisfying final step, let's put these pieces together and build our complete final map of this chromosome section.
So let's look at our 2 key findings side by side. The PAN gene is 32.7 units from the centromere, but the PDX gene is only 16.4 units away. This tells us the gene order right away, doesn't it? The PDX gene has to be closer to the centromere than the PAN gene is.
And here it is, our final map. We start at the centromere, we travel 16.4 map units to find the PDX gene, and then we keep going down the chromosome to find the PAN gene. But hang on, what's that last number? The distance between the 2 genes themselves? That last distance is just simple subtraction. The total distance from the start to PAN is 32.7. The distance to PDX is 16.4. So the distance between them has to be 32.7 minus 16.4, which gives us 16.3 map units. And that's it. We've successfully mapped the order and all the relative distances of both genes. So we just took a simple table of numbers and turned it into a detailed genetic map. This technique is absolutely fundamental to genetics. It allows scientists to find the exact location of genes that are responsible for all sorts of traits and diseases. It really makes you wonder, right? Now that we can make the map, what other genetic mysteries can we start to solve?